What happened here? Are you okay? You look upset.
See my crops? I have recently had a pest. All the effort and money I have invested are gone.
Haven't you thought about using pesticides?
Well, of course I have. But believe me, they are not the best solution.
Why not?
First, because they affect other species. Second, because you end up using higher doses. And third, they are not very well socially accepted and environmentally friendly, so I don't like them.
I see. Are there any alternative methods?
Well, yes, I have heard about the use of insect sex pheromones for mating disruption strategies.
Mating disruption? What is that about?
Oh, did you know that female insects lay eggs, release pheromones, then the male comes, follows their trace, they mate, and lay eggs on the plant. Then the larvae are born and eat the crops. So, if certain amount of pheromones are released into the air, then the male won't be able to follow the female's trace, so they won't mate. And the larvae won't be born, of course.
I see. That's quite an interesting technique. Why don't you use it?
Well, it's because nowadays the only way to obtain those pheromones is by chemical synthesis. And it needs a lot of specialized stuff, also installations to get rid of the residues. So at the end, it's very expensive, and I can simply afford them.
I see. Let me think. Hey, I have an idea. Imagine a plant able to protect crops by producing mating disruption pheromones when it the crop with affected plant species.
A plant? Look, these moths are eating my crops. Would you be able to produce a plant that releases insect sex pheromones?
I don't know. We can try.
Oh, and could that plant only work when the annoying pest comes?
It seems quite demanding, but we can try it too.
And that plant should be safe for humans and the environment as well.
For sure. We'll have a look at it.
If you get that, I take my hat off.
You look like an optimistic biotechnologist, but have you considered if the plant is biologically prepared to meet your needs? Or what about the dynamics of pheromone diffusion? Do you know how to distribute your plants in the crop field to cause mating disruption?
Surely, an ingenious perspective would be really helpful.
I can help you. Trust me, I'm an engineer.
Thank you. But such a demanding project requires gathering a multidisciplinary team. Let's go find them.
Let us present our project.
The sexy plant. The sexy plant rises as the newest pest management method. It is a safe, sustainable, and environmentally friendly alternative to current methods as pesticides or synthetic pheromones. Remember our three necessities. We need a plant able to produce the pheromones, able to release them, and safe. But let's start from the beginning. In order to produce moth sex pheromones, we need to reproduce their production pathway into our chassis, a plant called Nicotiana ventamiana. So, after an extensive research and based on previous publications, we established our biosynthesis pathway. These are our three objectives: 3 moth sex pheromones, based on a cysteine carbon fatty acid. One of them containing an alcohol group, another one being an acetate, and the third one being an aldehyde. Okay, but what do we have to start with? Our plant offers a cysteine carbon molecule that we can use as substrate for our pathway. Then, by adding 2 enzymes, a desaturase and a reductase from 2 different moth species, we can produce our first objective, our first pheromone, the alcohol. Then, the alcohol can take 2 different ways. It can be converted into the acetate by introducing an acetyltransferase from another plant, or it can be converted into the aldehyde by taking advantage of an alcohol oxidase from our plant, Nicotiana ventamiana. Therefore, summarizing, in order to produce more sex pheromones, we need to engineer a new metabolic pathway into our plant, Nicotiana ventamiana, comprising 3 different genes from moths and from another plant. Then we need to perform a multi-genic assembly in order to obtain a construct with the 3 genes suitable for plant transformation. To do so, we use a DNA cloning method called Giant Jamboree, which is especially adapted for plants. Then, we join a plant constitutive promoter, an enzyme code enricher, and a terminator into a single plasmid. We repeated this process with the 3 enzymes of our pathway and join them all together into a multigenic construct suitable for plant transformation. Alright, but however, our DNA cloning method, Giant Jamboree, and BioRich do not share the same standards. So, with the help of the NRP gene, we adapted our parts to BioRich and sent them to the registry. These parts were the 3 enzymes of our pathway. Alright, we finally have our multigenic construct, so we need to introduce it into our plant. To do so, we make use of a soil bacterium called Agrobacterium tunibesians, which, by literally injecting it into the plant leaves, is able to introduce in the plant cells the new genetic information and enabling the pheromone production. Well, is our plant really producing the pheromones? To know that, we perform an volatile organic compound analysis and perform a gas chromatography mass spectrometry. Alright, here are our results. But wait a second. Do you remember our pathway? We needed to produce 3 mold sex pheromones: the alcohol, the acetate, and the aldehyde. Alright, let's continue. This is how a normal Nicotiana mentamiana would smell like. Quite refreshing actually, but nothing related to our pheromones. And this is how the sexy plant smells like. Can you spot the difference? It's difficult to miss, right? That huge peak over there is the alcohol. Yeah, our alcohol. We not only made this plant to be the most abundant, we not only made this plant to be producing our pheromone, but also made it to be the most abundant to the plant. But wait, there's more. Can you see the other peak right there? This peak is the acetate. So it's not as big as the alcohol, but yet one of the most abundant compounds into the plant. In addition, we also produce aldehyde from the alcohol by means of an endogenous alcohol oxidase from our plant. So in conclusion, we obtained 3 different mold sex pheromones: the alcohol, the acetate, and the aldehyde. Alright, but we were much more ambitious. We also wanted to test the mold's pheromone interaction. So we made use of a technique called electrongenography. This technique works by connecting a mold antenna to 2 electrodes. Then an air current with plant volatiles is applied to the antenna. If among these volatiles are there mold pheromones, the antenna produces an electrical signal that can be detected, as you can see in this figure. Therefore, proving that there is interaction between our plant-produced pheromones and the moths. As you can see, we obtained very good results, but many more things needed to be considered.
For instance, an aspect that shouldn't be put away. What about metabolic burden? How much pheromone is our plant able to produce with existing substrate? Can we optimize our pheromones production? These questions could be answered by a mathematical approach called flux balance analysis. This is useful to analyze the fluxes through a metabolic network with constraint-based models. But plant metabolic networks are more complex systems than E. coli, so it is not that easy. Among the available ones, we selected the iGEM model. This is a model for functional primary metabolism of Arabidopsis thaliana. We know it is not exactly the same as our plant, Nicotiana mentamiana, but Arabidopsis is a model plant, and iGEM is one of the most complete models for plants by now. So then we implanted our metabolic pathway into the iGEM model, and we did the simulations in silico, with the aim of optimizing our pheromones production. Dealing with the optimization of our pathway substrate in terms of design and with the plant growth in biological terms, we found that these 2 objectives were conflicting. So we would have to look for a trade-off solution. The solution given by our coupled production yield increases with the photons uptake from the light. As you can see, FDA can help us to know our network production capabilities.
All right. Up to now, we have a sexy plant able to produce pheromones, but we needed more. We needed a plant able to release them efficiently only when necessary. In other words, we needed to control where and when the pheromones would be produced. In order to reduce the metabolic cost for the plant, increase the release ratio, and avoid uncontrolled pheromone release. So, regarding where, we focused on glandular trichomes. Glandular trichomes are a kind of hairs present on the plant surface and are specialized on releasing volatiles. So we cloned a glandular trichome specific promoter and tested its functionality, tuning it with GFP as a reporter. As you can see, it worked pretty well, and we have localized fluorescence on the head of the trichome, proving that we can target our biosynthesis pathway specifically on them. On the other hand, we wanted to control when the pheromones would be produced upon insect mating season. So we implemented our plan with a generic switch. This generic switch is based on a copper inducible promoter. So the plant needs copper sulfate as an inductor, which is inexpensive and commonly used in agriculture as a fungicide. Briefly, this switch works by producing a transcription factor that senses the copper ions concentration, changes conformation, and uses an operator in a chimeric promoter controlling a gene of interest. So, in order to test if it works, we use luciferase as a reporter under different copper sulfate concentrations, obtaining very promising results.
Well, how much pheromone is our plant able to release in reality? And how much do we need to get the result we want? It's evident that we need to model the pheromone diffusion mechanisms to predict how moths will respond. Pheromones are molecules that can undergo a diffusion process, moving away from its source. But diffusion processes are complex, so modeling them analytically and with accuracy is difficult. And even more when the geometry is not simple. For that reason, we decided to use a simplified model based on the heat diffusion equation. And then we use the envelope approximation to obtain a numerical solution. This solution gives us the temporal and spatial distribution of pheromone concentration in our field. Well, many moths seem to respond to pheromone concentration gradients, being attracted towards the source. This allows males to find the females, but we can disrupt male orientation by increasing the pheromone concentration up to unnaturally high levels. However, when one starts at moths, they apparently move with erratic flight paths. So we have chemotraction, random flight, saturation of the moth perception. How do we model these three things? Using a modeling environment called Logo, we simulated the approximate moth's behavior. When sexy plants are switched off, males move randomly until they detect pheromone traces from females, and in that case, they follow them. When sexy plants are switched on, pheromone starts to diffuse from them, rising up the concentration levels in the field. As the concentration rises, the insect perception becomes saturated, so it is less able to follow pheromone concentration gradients, and sexual confusion is induced. The parameters of this model are not as well characterized as we have expected at first. Big problem. So we decided to take an inverse engineering approach. We simulated many possible scenarios to come up with the values of the parameters corresponding to the desired one. Insects get confused. In qualitative terms, this would be useful to know the limitations of our system and helpful to decide the final distribution of our plants in the project.
And finally, the most concerning question. Is our plant really safe? Let's see. First of all, this plant is not going to be the consumed one. So this is a great advantage on biosafety. We also were concerned about this plant being spread without control into the field. So we made it sterile. We used an RNase taken from the PAR registry under the control of an anther-specific promoter. So, as anthers are the main part of the flower in charge of producing pollen, by expressing this RNA specifically on them, we avoid the plant reproduction. So, in addition, we were also worried about this plant being unequivocally identified in the field. So, we created an identity preservation module which induces anthocyanin accumulation. Anthocyanins are dark purple pigments So as you can see, we achieve a nice purple plant that can be easily identifiable in the field. So the answer to this question, is our plant safe? Yes, it is safe for living beings and the environment.
Wow, so we have designed a plant able to produce pheromones, lives under control and safe.
What an amazing work! I can't wait to see this plant on my field.
In fact, we know the protection of crops is essential for a social and economical sustainability in a world with an ever-growing population. Thus, we focus the whole project on the needs of our society. We consider the social, ethical, safety and environmental aspects that our project requires. In this sense, our work of policy and practices is not the cherry on top of the cake. It is the cake itself. Since the beginning, we have developed the sexy plan with the support and advice both from private enterprises like biocrop science and academics whose work is related with pest control. Also, we have interacted with farmer representatives to know the main involved people acceptance. They all got a good impression about the sexy plant and they are willing to see it.
That's great. I take my hat off.
To summarize, we have achieved a plant able to produce more sex pheromones. We have detected the interaction between these pheromones and the insects. We have analyzed in silico the pheromone production capabilities of our plant metabolic network. We have made 2 genetic modules to control the pheromone release, one for the trichome-specific expression and the other one for the switch. We have made a first version of a simulation environment for the pheromone diffusion and the moth's response. And we have made 2 genetic modules to make a completely safe plant, making it sterile and easily recognizable. We have interacted with relevant stakeholders. We have taken part in some collaborations with other teams. We have participated in the interplant study. And finally, we have sent our submitted notes. After this brief summary, we would like to remind you what our project consists of. The sexy plant is the newest pest control method. It is safe, sustainable, and environmentally friendly. For a long-term horizon, we would like to see this method being implemented in any plant, any ecosystem, and for any kind of pest. Thank you for your attention.
Thank you very much. That was really very impressive project. I'd like to ask you, is it important to have the particular ratio of different compounds in the pheromone to be effective? So to acetate, alcohol, and aldehyde, I think.